Hello everyone. Today we will discuss about vitamin B12 or cyanocobalamine. It is a water soluble vitamin. Synonyms are antipernicious anemia factor, antipernicious anemia factor, then extrinsic factor of Cyanocobalamine, and animal protein factor. These are the synonyms of vitamin B12. This is the structure of vitamin B12. It contains a central portion, that is cobalt atom. This cobalt atom is surrounded by 4 pyrrole rings. Here we can see 4 pyrrole rings. 1, 2, 3, 4. 4 pyrrole rings that are surrounding a cobalt atom. This ring is called, together it is called corin ring. Together it is corin ring. Below this corin ring is a 5,6-dimethyl benzimidazole moiety. 5,6-dimethyl benzimidazole riboside, that is called DB1 ring. DB1 ring. Then this DB1 is attached, one end of the DB1 is attached to the cobalt atom. Then next end is attached to a ribose moiety. This end is attached to the ribose moiety. Then one phosphate group is there. This phosphate group connects this ribose to the aminopropanol, esterified aminopropanol. This is then connected to the side chain of, this is connected to propionic acid. Then that is connected to the 4th chain of this corin ring. So it contains 4 pyrrole rings, then cobalt atoms, then DB1 ring, that is 5,6-dimethyl benzimidazole moiety, then ribose moiety, phosphate, then propionic acid and aminopropanol. This is the entire structure of vitamin B12. Then a cyanide group is attached to the cobalt atom and it is called cyanocobalamine. If cyanide group is there, it is called cyanocobalamine. If cyanide group is absent, it is called cobalamine. This is the entire structure of vitamin B12. Next we will see types of vitamin B12. When cyanide group is bound to cobalt atom, it is called cyanocobalamine. If cyanide group is removed, then it is cobalamine. Then hydroxyl group, then NO2, chloride, sulfate may replace cyanide group. So types are hydroxycobalamine, that is B12A. So it contains hydroxyl groups. Next is nitritocobalamine, or B12C. It contains NO2 group. Next one is chlorocobalamine. It contains chloride group. Then sulfatocobalamine. It contains sulfate. Then in this, hydroxycobalamine or B12A is more active in enzyme systems. It is retained longer in the body when given orally. So B12A is more useful for therapeutic administration of B12 by mouth. That is the more useful, this is the more useful one for therapeutic administration. Next we will see metabolism. First one is absorption and excretion. Absorption and excretion. Vitamin B12 is absorbed from ileum. It requires presence of HCl and intrinsic factor of Kcl. This is a constant of normal gastric juice. So, for the absorption, it needs presence of HCl and intrinsic factor of Kcl. This is a constant of normal gastric juice. Intrinsic factor means it is a glycoprotein. It is a glycoprotein. It is secreted by parietal cells of gastric mucosa. Intrinsic factor, parietal cells of gastric mucosa. It is a glycoprotein. It can be destroyed by heating at 70 to 80 degree for half an hour. It is non-dialyzable and thermolabile, but it can be destroyed by heating at 70 to 80 degree for half an hour. Then it can be inactivated by prolonged digestion with pepsin or trypsin. It is found in cardiac end and fundus of stomach. That is intrinsic factor. Next we will see mechanism of absorption. Mechanism of absorption. Two binding proteins are required for absorption. Two binding proteins, they are cobalophyllin and intrinsic factor. Cobalophyllin and intrinsic factor. This cobalophyllin is a binding protein. It is secreted by saliva, secreted in saliva. Then intrinsic factor is a glycoprotein secreted by parietal cells of gastric mucosa. These are required for the absorption. These are two binding proteins. Then HCl and pepsin release vitamin B12 from protein binding in food, and it binds to salivary protein cobalophyllin. HCl and pepsin release vitamin B12 and this vitamin B12 binds to this cobalophyllin. Then in the duodenum, cobalophyllin is hydrolyzed and release the vitamin for binding to intrinsic factor. In the duodenum, it is hydrolyzed and it will release vitamin B12 to bind to the intrinsic factor. Vitamin B12 is absorbed from the distal third of ileum through specific binding site or receptors that binds B12 intrinsic factor complex. Then from the complex, B12 enters the ileal mucosal cells for absorption into the circulation. That is the mechanism in the absorption. Next is transport in blood. Transport in blood. It is transported in blood in association with specific proteins called transcobalamine 1, 2 and 3. Transcobalamine 1, 2, 3. Physiologically, 2 is more important. Transcobalamine 2. So, the vitamin B12 is transported in blood in association with specific proteins. These proteins are transcobalamine 1, 2 and 3. This 2 is more important. This 1 is increased in transcobalamine is increased. 1 is increased in myeloproliferative states. Myeloproliferative states. Then its source is leukocytes. Plasma level is about 60 μg per liter. Then coming to 2, transcobalamine 2, its plasma level is 20 μg per liter. Its source is liver. That is the transport in blood. Normal serum level is about 0.008 to 0.42 μg per dl. 0.008 to 0.42. Coming to excretion, there is no urinary excretion, but following parenteral administration, there is urinary excretion up to 0.3 μg per day. That is excretion. Then coming to storage, main storage site is liver. This is the main storage site. This is the metabolism. Next one is biological active forms. Biological active forms. Biological active forms of vitamin B12 are cobamide coenzymes. Cobamide coenzymes. They act as coenzyme with various enzymes. This cobamide coenzyme do not contain the cyano group attached to the cobalt, but instead there is an adenine nucleoside. So we will see the cobamide coenzyme. CHOH, again CHOH, CH, then CH2, here cobalt. So this cobamide coenzyme do not contain the cyano group attached to cobalt, but instead there is an adenosine nucleoside. It is contain adenosine nucleoside or adenosine 5' deoxynucleoside. 5' deoxyadenosine. This is 5' deoxyadenosine. This is linked to cobalt by a C-CO bond. That is cobamide coenzyme. This 5' adenosyl moiety is derived from ATP. This is derived from ATP. In the formation of adenosyl coenzyme, cobalt undergoes reduction in a series of steps. These are catalyzed by the enzyme B12A reductase. B12A reductase. So in the formation of adenosyl coenzyme, this cobalt undergoes reduction in a series of steps. These are catalyzed by the enzyme B12A reductase using NADH and FAD. Then this B12 cobalt reacts with ATP to form adenosyl coenzyme. Adenosyl coenzyme. Then we will see types of cobamide coenzymes. Types of cobamide coenzymes. First, four types are there. First one is DBC. It contains 5,6-dimethyl benzimidazole. It is called dimethyl benzimidazole cobamide. This is dimethyl benzimidazole cobamide. That is DBC. Second one is BC. It is benzimidazole cobamide. Benzimidazole cobamide. Third one is AC. It is adenyl cobamide. Adenyl cobamide, which contains an adenyl group. Then fourth one is MC or methyl cobamide. Methyl cobamide. In this CH3 group is attached to cobalt atom. Methyl group is attached to cobalt atom rather than adenosyl moiety. Then these coenzymes do not contain the cyanide group, and so they are called corinoid coenzyme. These do not contain cyanide group and they are called as corinoid coenzymes. Corinoid coenzymes. These are the types of cobamide coenzymes. Next we will see metabolic roles of cobamide coenzymes. Metabolic roles of these cobamide coenzymes. First reaction is methyl malonyl CoA to succinyl CoA. Here B12 act as the coenzyme. That is L-methyl malonyl CoA is converted to succinyl CoA. Here B12 act as the coenzyme. Enzyme is isomerase. So in B12 deficiency, methyl malonic acid accumulates, and so excretion of methyl malonic acid in urine increases. So methyl malonic aciduria is a sensitive index of B12 deficiency. Due to if B12 deficiency is there, it accumulates and its excretion in urine also increased. So methyl malonic aciduria is a sensitive index for B12 deficiency. Next metabolic role is conversion of methylation of homocysteine to methionine. Homocysteine to methionine. Here this requires FH4 tetrahydrofolate as a methyl carrier for the conversion of or methylation of homocysteine to methionine. Next reaction is methylation of this pyrimidine ring to form thymine. Pyrimidine to thymine. Methylation of pyrimidine to thymine. Next one is conversion of ribonucleotides to deoxyribonucleotides by the enzyme reductase. Ribonucleotides to deoxyribonucleotides by the enzyme reductase. Here also vitamin B12 act as the coenzyme. Next one is required for metabolism of diols. Metabolism of diols. For example, ethylene glycol, CH2OH and coenzyme B12, it is converted to acetaldehyde, CH3CHO plus H2O. So for the metabolism of diols also, this vitamin B12 act as the coenzyme. Then in bacteria, interconversion of glutamate and beta methyl aspartate. For the interconversion of glutamate and beta methyl aspartate, that is glutamate or glutamic acid by the, here coenzyme is B12, by the enzyme mutase. Glutamic acid is converted to beta methyl aspartic acid. Beta methyl aspartic acid. This is then converted to mesaconic acid. Mesaconic acid by the enzyme beta methyl aspartase. Beta methyl aspartase. So this is in bacteria. For the conversion of glutamate to beta methyl aspartate, B12 act as the coenzyme. So these are the metabolic roles. Next we will see methyl trap hypothesis. Methyl trap hypothesis, otherwise known as folate trap. That is N5, N10 methylene THF is converted to, by the enzyme reductase, it is converted to N5 methyl FH4. N5 methyl FH4. Then this is with the help of B12 as the coenzyme, it is converted to methyl B12. Methyl B12 plus FH4. FH4 to 1 carbon metabolism. Then this methyl B12 is converted to, is required for the conversion of homocysteine to methionine. For the conversion of homocysteine to methionine by the enzyme homocysteine methyl transferase. Homocysteine methyl transferase or methyl synthetase. Methyl synthase. For this is called folate trap. Let's see it's in detail. B12 is necessary for the conversion of N5-methyl FH4 to form methyl B12. Here B12 acts as the coenzyme. This methyl B12 is required for the conversion of homocysteine to methionine. So, in B12 deficiency, this reaction cannot take place. So, this N5-methyl FH4 cannot be converted to methyl B12. So, this folate is permanently trapped as N5-methyl FH4. In B12 deficiency, this conversion does not take place. So, folate is trapped in the form of N5-methyl FH4. So, it is known as folate trap. So, it is not available for the C1 metabolism, 1 carbon metabolism. So, it is called folate trap. This results in diminished synthesis of thymidylate and DNA. Increased folate levels are observed in plasma and the activity of the enzyme homocysteine methyl transferase is low. That is called methyl trap hypothesis or folate trap. Next we will see deficiency manifestations. Deficiency manifestations. First one is adult pernicious anemia. Adult pernicious anemia. This vitamin B12 deficiency produces macrocytic anemia. Macrocytic. It is characterized by hematological and neurological features. It is an autoimmune disease. It shows mucosal atrophy of stomach, then glossitis, stomatitis and pharyngitis, then echlorhydria, degenerative changes of posterior and lateral columns of the spinal cord, then it results in ataxia and paralysis. This is caused by the increased concentration of methyl malonic acid. Increased concentration of methyl malonic acid, which competes with malonyl-CoA, leading to impairment of fatty acid synthesis. Is it due to increased concentration of methyl malonic acid, which competes with malonyl-CoA, and it leads to impairment of fatty acid synthesis. Then hematological changes include macrocytic anemia, granulocytopenia, thrombocytopenia and reduction of RB cell count and hemoglobin count. Then coming to biochemical changes, which include decreased serum B12, decreased serum B12, decreased urinary B12, then increased excretion of fecal B12, then increased urinary excretion of methyl malonic acid. These are the biochemical changes. Then bone marrow shows evidences of arrested development of all elements, that is erythroid, myeloid and thrombocytes. Then megaloblast and myeloblast accumulates and number of megakaryocytes decreases. These are in bone marrows. Then in pernicious anemia, DNA synthesis is also depressed. This is adult pernicious anemia. Second deficiency is congenital pernicious anemia. Congenital pernicious anemia. It occurs in postnatal period, before 2.5 years of age. Before 2.5 years of age. It is due to lack of intrinsic factor. It is not accompanied by gastric acid secretion. Third one is juvenile pernicious anemia. Juvenile pernicious anemia. It is due to failure of secretion of intrinsic factor. Failure to secrete intrinsic factor. It is associated with echlorhydria and atrophic gastritis. Then absence of this intrinsic factor or parietal cells are demonstrated. There will be failure to secrete this intrinsic factor. So absence of intrinsic factor will be there. Then next fourth one is following surgical operations. It is seen in total gastrectomy, then extensive resections of small intestine. Next one is tapeworm infection in GI tract, tapeworm infection of GI tract. So these are the deficiency manifestations. Next one is sources. Sources of vitamin B12. It is present in foods of animal sources only. Animal sources only. It can be synthesized by intestinal bacteria. Rich sources are liver, egg, fish, meat and kidney. Fair sources are milk and dairy products. Daily requirements for adults 3 μg per day. 3 μg per day. Then for infants it is about 0.3 μg per day. Then for children it is about 1 to 2 μg per day. This requirement increases in pregnancy and in lactation. So this is about vitamin B12. This is today's topic. Thank you for watching.